Hello, everybody. Welcome to Silence Abounds Science at Home, where we will discover home lab magic. We will perform experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and even forensic science. Welcome to my channel. Hello, everybody, and welcome to Silence Abounds Science at Home, where we will discover home lab magic. I'm excited for today's experiment. We're going to look at the oxidation of an aldehyde in order to make and produce a mirror surface on a petri dish. We're going to look and see how mirrors are made and formed. Mirrors have been used for thousands of years. A mirror is an object that does not allow light rays to pass through it, but instead reflects them back. Silver, being very shiny, reflects light well and is used often to make mirrors. And that's why mirrors are expensive, because of the use of silver. When aqueous silver nitrate is mixed with aqueous ammonia, it forms a silver ammonia complex. And this is known as Tollin's reagent. Tollin's reagent is a chemical reagent used to distinguish between aldehydes and ketones. It exploits that the aldehydes are readily oxidized, whereas ketones are not. In this experiment, we will use a redox reaction between the silver ammonium complex ion and the aldehyde sugar glucose. The glucose acts as a reducing agent to reduce the ammonium complex ion to produce metallic silver, which plates out on a transparent surface and forms a mirror. We're going to use a bottom of a petri dish as our transparent surface. For this experiment, we are going to need a 2.5% silver nitrate solution. I've already made up the solutions ahead of time in the interest of time, but please note, when you're handling silver nitrate solution, you need to wear gloves, because silver nitrate is not a pleasant compound and it will stain your skin and it will stay stained for a very long time. So you don't want to get it anywhere on your skin. And for this experiment, you should wear gloves at all times, and I will do so in just a moment. In addition to the silver nitrate solution, you need concentrated ammonia. You need a 4.5% potassium hydroxide solution, which again, I've made already ahead of time. And you need a glucose solution, which again, I've made. And what I did with that is it's 2 grams in 20 mLs of water. And to that, I added ethanol about 2.5 mLs of ethanol to the solution, and I put it into a spray bottle. And you'll see the reason why later. So I've got my solutions all ready to start the experiment. You'll also need a cold bath, an ice bath. So I just got a large beaker and I put some ice in it. And you need a couple of beakers and measuring cylinders as well for the experiment. With that, let's begin. Okay, first things first is I'm going to put my gloves on. Remember, whenever handling silver nitrate, you want to be wearing gloves at all times. So I'm just going to put my gloves on. And then what I'm going to do is I'm going to take a beaker, just a 50 ml beaker. And what I'm going to do is I'm going to pour 13.5 mils of the silver nitrate into that beaker. So I got a measuring cylinder and I want 13 mLs. So that's 10 mLs. And I want 3.5 more. Okay, it's a little bit much. Okay. So I'm just going to wipe the outside so there's no liquid that I accidentally touch. Okay. I'm going to put that to the side. Next, I'm going to add the concentrated ammonia to the solution, and I'm going to it's going to precipitate, form a precipitate, and then I'm going to add the ammonia drop by drop. Like so, and I'm going to mix it. And then what we're going to do is I'm going to continue adding the ammonia until the solution clears. Okay, so I'm going to keep adding ammonia and stirring in between the addition of the ammonia. So add some more. And when handling ammonia, I would advise handling it in a well-ventilated area. You could do this experiment outside or in your garage. So I'm going to keep mixing it. And then I'm going to add more. I'm going to keep adding, and eventually this will clear. It take a little bit of time. So I'm going to add more. When you're mixing, try not to spill anything or splash anything, any of the solution. You don't want that to happen. Always have some paper towels nearby you in case of any splatters or spillages. Okay, so I'm going to add some more. And it's starting to clear. Okay, so it's gone clear. I mean, there's a tinge of color behind, but for all purposes, the precipitate has gone and the solution is clear. So I'm going to just add 3 extra drops of ammonia. 1, 2, 3 to the beaker. And as you can see, it is now completely cleared. And that's what we wanted. So now, what we want to do now is we want to add 3.4 mLs of potassium hydroxide to the beaker. And I'm going to use a pipette on that, a 1 ml transfer pipette. So there's graduations on these pipettes, and one full length of the pipette is 1 ml. So I need 3 of those. 1, 2, 3, and 0.4. Okay. So we formed another precipitate has formed. So a dark brown precipitate has formed. So now what we want to do again is we want to stir in some ammonia again, drop by drop. Okay, we're going to do this very slowly at this stage. So going to do it drop by drop, again mixing it in between the addition of your ammonia. And we want to do this again until it goes clear again. And it's starting to clear. I can see a change. Okay, now I'm going to add a little bit more. Okay. Add some more. And then we're going to add 3 more drops. 1, 2, 3. And now it's all gone clear. Okay, now we repeat and we add another 3.4 mLs of potassium hydroxide. 1, 2, 3, 3.4. Okay, 3.4. We give that a mix. All right, and then we're going to add several drops of ammonia to the solution. And now what we're going to do is we are going to place the beaker into our water our bath, cool bath, and we're going to let that sit until it's cooled down to about 15 degrees C. Okay. Okay, so the solution's been sitting in our ice bath for several minutes, so I'm just going to check and verify the temperature because I want it down to 15 degrees. So I'm going to put this in the solution and see what temperature it is. And so it's reading around 15, 16 degrees, which, like I said, is what the temperature of the solution I wanted it to be at. So let me put my thermometer away. Okay. So next what I want to do is I'm going to pour that solution into my petri dish, but I'm going to do it in this box, and I'll explain why in just a moment. So there's nothing to see right now, so I'm just going to put the petri dish in this box, and then I'm going to pour my solution from this beaker into the petri dish, and you want to make sure that the whole of the petri dish is covered by the liquid. So you can just move it about just to equilibrate and make sure everything is covered, the surface is covered. So now we're going to do the reaction, okay? And the glucose solution is in a spray bottle because I'm going to spray and create a mist of glucose around the petri dish. I am not going to spray directly into the petri dish. I don't want that. In order for the reaction to take place, you want to create a mist. So I've got my bottle, my spray bottle, and I'm going to just lift this box up like so, just to try and contain the mist vapors. And I'm going to spray about 3 mLs of the glucose solution over the petri dish. But I'm holding the spray bottle so it sprays horizontally. Okay, I don't want the spray going into the petri dish. So I'm creating a mist like so. And I don't want it going into the petri dish directly. So I'm creating a mist. And I need about 3 mLs of this to work. All right, that should do it. So now we let the redox reaction take place, and this is going to take about 10 to 15 minutes. So we'll come back when the reaction is complete. Okay, it's been about 20-30 minutes that I left the reaction to fully react. So let's see whether it worked. Here is the big reveal. So, oh, yes. So I'm going to gently remove the petri dish out of the box. And then I'm going to try and remove the liquid from the petri dish. And what we have is a mirror surface on the bottom of the petri dish. I don't know if you can see this, but we have formed a mirrored surface from the oxidation of an aldehyde. So here I'm doing a close-up, and you should be able to see the reflection of me videoing the mirror surface. And this reaction has worked beautifully. Well, I hope you enjoyed that experiment. It took a little bit of preparation, and reaction to occur took a little bit of time, but the result was a very interesting result, and it's very exciting after an experiment that you put all the effort into produces the results that you're expected, and that was clearly indicated today. So let's recap and let's revisit the whole reaction. So an aldehyde is a common functional group in organic chemistry. Aldehydes are responsible for natural and synthetic hormones. The structure of an aldehyde consists of a carboxyl group single bonded to a hydrogen atom. Aldehydes and ketones are very similar in structure. They both contain a carboxyl group and a double bond to oxygen. The main difference between aldehydes and ketones is the position of the carbonyl group in the molecule. Let's talk about redox reactions, oxidation, and reduction. When oxidation occurs, there usually is a loss of electrons. You get an increase in the oxidation state. So in this experiment, the carbon in the aldehyde group goes from gains a hydrogen and goes from a +1 to a +3, but it loses 2 electrons. So that's the oxidizing reaction, the oxidization of the aldehyde. Also, in oxidation, it often involves gaining oxygen or losing hydrogen. In reduction reaction, when the compound is reduced, it's the opposite. Compounds gain electrons. So in our experiment, the silver ions are reduced, going from a silver Ag plus to an Aj zero. So they gain electrons and they gain and they decrease to an oxidation state, going from +1 to 0, and gains 1 electron. And also in reduction reactions, they are often involved gaining a hydrogen or losing oxygen. So it's the complete opposite to oxidation. And there are many different types of experiments that demonstrate the principle of redox reactions. And we're going to be doing many more in the future because it's a very, very important fundamental concept in chemistry. So we will go into more depth into redox reaction in future videos. So the overall redox reaction of today's experiment was that the aldehyde oxidized to carboxylate and silver ions reduced to metallic silver. Well, I hope you enjoyed that video and that experiment. Please subscribe to my channel. It's free of charge. Please hit the like and share buttons and feel free to leave me a comment. Please also check out my Patreon page where I publish exclusive membership videos that will not be shown on YouTube. I also publish all the recipes to all my experiments along with video background notes of the experiment. Again, these are all published for membership on my Patreon page. Please feel free to email me, send me your questions, your comments, send me your photos and videos of you doing your experiments in your own home. I love to share and build up a community. Check out my resources in my description of the video. I list all the resources that I use to purchase kits, experiment kits, lab equipment, glassware, chemicals, etc. Everything you need to do home experiments. Until next time, keep on experimenting. Goodbye, everybody.